Hi, my name is Allie Slater.
And my name is Raichel, and Allie and I are both current sophomores at Redwood High School. This year, we performed an experiment that we entered into the Redwood High School Science Fair. Our experiment tested the effect of pesticide concentrations on the genetic variation and health of strawberries and their plants.
Driving around in Marin County, you will see the Priuses and the recycle signs, which are all examples of the green movement. Growing up in this environment, both Raichel and I have been exposed to this idea of the green movement, which involves the advocacy and production of organic foods. Basically, adults in Marin will shop at their local markets such as Whole Foods, Trader Joe's, and Paradise Foods in search of products untreated by pesticides, or in other words, organic.
But what even are pesticides? Pesticides themselves are chemicals that protect plants from diseases and pests. As plants are protected with pesticides, they are more likely to survive production, which therefore reduce their costs. With reduced costs, they are then often sold at supermarkets like Safeway and Costco that appeal more of the general population compared with the markets Allie mentioned.
However, recent studies have shown that pesticides may have negative impacts on the human body, being that they can be carcinogenic. Our goal with this experiment was to head straight to the source and discover if the pesticide influence has any negative impact on the plant itself, rather than the human body.
Limited research conducted on the plant itself has shown that pesticides increase the stress on plants and therefore negatively impact the plant. We transferred this information to form a hypothesis. We hypothesized that there would be a direct correlation between the pesticide concentration and genetic variation.
This research provided incentive for our experiment, in turn assisting in the development of our procedures.
In our procedures, we purchased and applied Ortho Insect Control, which is a pesticide sold commercially worldwide. We apply this pesticide to 12 untreated plants using 4 different concentrations at 0%, 2%, 4%, and 12%. Usually, Ortho Insect Control is applied once per growing season, but what we wanted to do with this experiment was to test the effect of the pesticide over many growing seasons versus just one. With this in mind, we applied the pesticide on a weekly basis while we grew the plants.
Over the course of 3 summer months, Raichel and I applied the pesticide to the plants in a monitored and controlled environment. At first, the plants were similar in appearance, virtually indistinguishable.
However, after about 2 weeks, we started to notice distinct differences among the plants. For example, the plants applied with higher concentrations of the pesticide provided less yield and were typically smaller in size.
Additionally, something unexpected occurred. The strawberries, which were treated from the highly concentrated pesticide influence, were extremely deformed. In addition to being smaller, parts of the strawberries were cratered and discolored.
Besides appearance, though, the 4% and 12% concentrations also had a much harder time absorbing the water into their roots. This was demonstrated by the time it took to absorb the water. For the plants in the 4% and 12% concentrations, it took nearly 1.5 minutes, which was 3 times what it took for the organic and 2% concentration plants.
With that in mind, we decided to analyze and get a clearer picture of our data through a process known as biomass index. Biomass is the total mass of an organism in a given area or volume without the presence and in the absence of water. With strawberry plants, you first strip the plants of its leaves and dehydrate the roots. Then the final mass of the dehydrated roots will show how the plant was able to build root and plant stem material. This correlates directly to the pesticide's effect on the plant.
Another technique we decided on to use to test the effect of the concentration was gel electrophoresis. To use gel electrophoresis, it is essential that the material used that you extract the DNA out of is accessible and relatively simple, which is why we chose the strawberry plant.
To extract DNA from a strawberry, various substances are used to break down the cell and nuclear membranes. Once this is done, the DNA is exposed and can be isolated by exploiting the characteristics of DNA, which include its insolubility in alcohol. After the ethanol alcohol was added, the DNA precipitates and can be manually extracted.
As the DNA has now been isolated, it can be put through a few steps to prepare it for the gel machine. The gel electrophoresis machine includes an agarose gel, which has the consistency a little thicker than Jell-O. It is in this gel that the DNA solution will separate based on the length of the DNA strands in the solution when the electricity is added. The DNA will appear in bands based on the length of the fragments. The longer fragments will travel shorter because they can't travel through the gel as quickly, and the shorter fragments will travel farther. As a result, this will be different based on how damaged the plants are.
The data collected based on the observations recorded at the end of harvesting showed a trend that health worsened as pesticide concentration increased. This was true in terms of density, yield, and deformity. In particular, biomass revealed that, as expected, the control had the greatest final biomass of roots, suggesting that it was the most developed and most favorable.
However, this was not true of all the tests. In fact, in the density, yield, and deformity, the 2% concentration actually proved to be the most successful. For example, in the yield, in the 2% concentration, there grew an average of 6 strawberries, when compared to the organic plant that only grew 5. Since pests were not a factor here, nor was disease, the only variable was the concentration. This indicates that perhaps some limited pesticide can actually increase the production and overall health of the strawberries.
Although we collected many successful test results, the use of gel electrophoresis did not result as expected. Hypothetically, under UV light, the DNA fragments and the variations would be visible.
Unfortunately, ours showed none. This could be due to a variety of reasons, including human error and unreliable procedure sources. The most likely cause, though, is the natural degradation of DNA. Over time, DNA naturally breaks down, especially with freezing and thawing. In our experiment, every time we picked and harvested a strawberry, we would freeze it and then take the other strawberries out to compare it to that one we just put in the freezer, making this characteristic a likely source of error in our lab.
In order to conduct our experiment, we also had to wait 2 months until all the strawberries had grown and been harvested. In this time period, the DNA could have degraded further than it had already done due to freezing and thawing of the strawberries.
But there's another option for the source of error. It is possible that the error came in actually running the gel. Assuming that the DNA wasn't too damaged to run through the gel, it is possible that the DNA was so fragmented that the DNA ran off the gel when the electricity made it move. When running the gel machine, the loading die is placed into a DNA solution with the DNA to give an estimate where the DNA will be in the gel once the electricity is added. This allows viewers to stop the electricity before the DNA actually goes off the gel. If the DNA is too broken, however, the loading die won't even help.
We can assume that it is likely a combination of many of these errors. Additionally, given that this was the last test performed, we can assume that all of our other tests were quite accurate, especially given that we had to wait a long duration before we could perform this test in comparison to the others.
To further analyze our collected data, we used inferential statistics, including the ANOVA and t-test. In the ANOVA, entire sets of data are analyzed based on their variation in relation to the other data in that set. Similarly, the t-test also compares the data in the same way, but only compares 2 variables, like the 4% and 12% concentrations. Both of these tests allow scientists to determine the statistical significance or accuracy of their data. In a good set of data, 95% accuracy is favorable.
In our ANOVA test, the F calculated value was greater than the F critical value, which shows that our data was significant. This test also shows that only 4.6% of the values occurred by chance, supporting the idea that this test could be repeated and replicated to similar results. This, along with the t-test showing similar data, disproved our null hypothesis, which stated that the pesticide concentration would have no effect on the genetic variation or health of a strawberry plant.
Overall, the trend seen in our data suggests that with the exception of the 2% concentration, the increased pesticide concentrations reduced health and increased genetic variation in strawberries and their plants. This actually supports what pesticides are supposed to do. Many pesticides today, in addition to killing plants, have been known to increase productivity in plants.
From the results Raichel and I found in our report, it seems that the 4% and 12% concentrations were simply over-applied, with pesticides to the point where production rates and size dropped drastically, as mutations likely increased considerably.
In comparing our results to previous studies done, our yield exemplifies a previous one done at Dartmouth College. Their study showed that the stress reduces mitosis rates, which therefore lowers yield. This in turn supports our lowering yield overall, especially in the higher concentration pesticide plants.
As the biomass dry weights declined because of the pesticide increase, it was clear that the root size and quantity contributed to the health of the plant. This could also reduce yield, as the plant was receiving fewer nutrients. But what does this mean for society today?
Right, so as we brought up earlier, many people debate whether organic foods are worth the cost. In terms of health, organics seem unnecessary unless the pesticide is at a very high concentration, like our 4% or 12%. The high concentrations in particular were deformed, which shows us that at some point pesticides do affect the plant itself. At this moment, the degree of pesticide application is such that we have no means to fear concern for the plant's health.
Therefore, our experiment successfully determined the insignificant impact of controlled, limited pesticide influence on strawberry plants in comparison to organics.
Thank you.
All right, we're going to have to cut that.